And it is now my pleasure to introduce the next speaker in this session, who is Bartha Knoppers. Bartha leads the Center for Genome Policy at McGill University, and she is also the chair of the Human Cell Atlas Equity Working Group. And I will hand it over to Bartha.
Hello, everyone. It's a pleasure to be here. And thank you, Jonah, on behalf of all the researchers involved in the HCA for this announcement. It's really inspiring, and I'm very glad it's before our session on pediatrics. Next slide. As you know, and you can see from the table of contents of my presentation, the areas to be covered have always been contentious ones. And in fact, there was a time when you could not even do research at all on embryonic tissue. Pregnant women or potentially, or women of reproductive age or potentially pregnant could not be included in clinical trials. Fetal tissues could not be used, and so on. And children themselves, it wasn't until really about 20 years ago, and even more recently under the European Clinical Trials Directive, where it explicitly states that you must include children in research, because there are conditions and knowledge that we are missing that is particular to this population. Next slide. So I'm legally trained, if you haven't or aren't aware, and you'll see that most of my slides center around a human rights approach to discussion of these issues surrounding fetal, embryonic, and pediatric research. So I'll start with, I think, the most beautiful statement that dates back to the Universal Declaration of Human Rights, and that is that all human beings are born free and equal in dignity and rights. They are endowed with reason and conscience. And what's interesting here is that it has often been missed that you are free and equal in dignity as a human being. So it's inherent in the human being. And because of that, we have the creation of human rights. So dignity is not an ordinary a human right. It is actually inherent in human beings, and thus we create human rights to respect that dignity. Next slide. There's only one slide on this contentious issue, which is and will be probably due to social, cultural, and religious differences around the world, a lack of consensus, if you like, on the nature and status of the embryo, the fetus, and so on. And this comes from a decision of the European Court of Human Rights 2004, where the court stated, at best, it may be regarded as common ground between the states, so different countries, that the embryo and fetus belongs to the human race. The potentiality of that being, and so human being, and its capacity to become a person require protection in the name of human dignity without making it a person with a right to life. So you can see how the notion of being a legal person, recognized and protected as a person, which can be whenever a state declares it to be, a born, alive, and viable, for instance, used to be in the 1804 French Civil Code. It doesn't mean that human beings themselves are not protected under different kinds of legislation, even though they are not yet legal persons. Next slide, please. So let's get right into the issue. Research with embryonic tissue, I can't believe it, but I calculated it was 32 years ago that the 14-day rule was accepted following the Warnock Commission report in the UK. And don't forget, Louise Brown was born in 1978, first in vitro fertilization baby. So that 14-day rule on the number of days that research could be done in those countries that decided to adopt laws to that effect, or regulations, or professional standards, or human fertility embryology agencies that would decide, that 14-day rule has stood firm where research is permitted. But 2 months ago, even though it is not technically feasible to culture human embryos beyond formation of a primitive streak or 14 days post-fertilization. However, culture systems are evolving, making this a possibility in the near future. Well, that was the International Society for Stem Cell Research, ISSCR. That was a new revolutionary statement. They did, however, confirm that any use of embryos, fetal tissues, or other cells or gametes and so on should be subject to the rule of an fully informed and free consent. Again, in August 2021, so a month ago, the French law on surrounding assisted procreation also confirmed the consent rule for donation of tissue. Next slide. Nevertheless, it didn't take long as far as the ISSCR guidelines were being discussed. First of all, to explain why the ISSCR guidelines needed an update. But immediately, scholars or colleagues in the field of ethics, law, and society, and LC scholars said that this omission of an age limit, saying that the 14-day rule could be extended, perhaps, it would be a case-by-case determination under a regulated, governed structure as permitted or not in different societies. And this was determined to be a grave omission. Everybody, not everyone, but persons in this article thought that the certainty that the 14-day rule offered was what was needed in this area of research. Well, in contrast, myself and a number of other scholars thought that this more contextual case-by-case evaluation and governance, if the science was of quality and it was safe and it was efficient and it could really further our understanding of the role of embryonic development and human development generally, that this particular opening, small as it is, actually upheld another human right, and that is also found in the Universal Declaration and in an international covenant signed and ratified by 179 countries, saying every human being or everyone has the right to benefit from science, from scientific progress, from advances in science, and that extending this rule had incredible implications for understanding miscarriage, genetic origins of certain conditions, and so on. And we thought that this was an outstanding example of finally bringing this right of citizens to benefit to the fore. Next slide. So a little bit closer, specific to fetal tissues as opposed to embryos or to other tissues where usually the consent of both donors or parents are concerned. In fetal tissue, again, since the 90s and 80s and 90s, embryos and so on will require a voluntary informed consent. But in the case of fetal tissue, consent from the woman donating the tissue is sufficient. Now, obviously, I'm talking about countries where research with fetal tissue is allowed, but this is a distinction, seeing as it is part of the woman in terms of not only her body, but her human rights to decide what can or cannot be done with fetal tissue. Next slide. Now, if that's all very clear, here is a map that we did about 5 years ago, looking at research guidelines on different areas around the world with categories of restrictive, intermediate, permissive, forbidden, regulated, and so on. And you'll see there's quite a difference between different countries with respect to some of these areas of research. And we know how fast that can change, and we also know how the inability of some countries to enforce their laws, reinforce their laws, can also lead to difficulties in terms of professional integrity. Next slide. So turning then to the child and leaving the fetus and the other tissues for embryos and so on for the time being. Children, as you know, have their own convention. It took 10 years, like most conventions do, to make it to be adopted. And it's universally signed and ratified except for one country. And children have the right to their interests being primary consideration in all decisions concerning their well-being. They have a right to be heard and the right to the highest attainable standard of health. So those are their rights. Parents have duties and rights as well under law and also ethics guidance. They include the right to, or the duty to provide medical care and the authority to involve their children in biomedical research. So they have that freedom to do so. There's not an article on research on children in the convention, however. They were not able to come to agreement on that. But there's above the parents, there's also the state. The state has, under what's called a parents' patriae doctrine, the ability also to protect children over and above parental protections and duties, which they do through child protection legislation. And they can also, in certain cases, be overruled by the state, like in vaccination and so on. Next slide. Biomedical norms, give an example here of genetics and genomic research, where even if parents don't want to know following genetic testing, if a condition is actionable during childhood, can be prevented or treated, most European countries and Canada, for example, will say that that information should be given and the child should be treated. And there's also examples of parents being able to participate in research, and I put a clause there that you find in most forms. Next slide. So behind all of this is the right to science, which I already mentioned, which obviously applies to children as well, the right to benefit from science and the freedom of science as well. The right to health, which is in the Children's Convention, but also in other conventions, and the right to non-discrimination. We heard in an earlier session today about the equitable distribution and benefits of in the health sector. Next slide. So are there any legal limitations? Well, of course, some states can prohibit or they can allow certain research to go ahead, but you can only limit human rights. So that's different from your ethics documents or your local laws. You can only limit human rights by law, and only insofar as those limitations are compatible with the nature of those rights to health, to science, and so on, and solely for the purpose of promoting the general welfare in a democratic society. So when you see these rights, they give us the potential to not only realize them and give them meaning, but also to perhaps even one day determine future rights as well, or come out with future rights for children in this area. Next slide. But what if you have rights in conflict? Parents' rights of religious freedom with the child's rights under the convention. Well, you have to balance these in a proportionate way, these rights and freedoms, subject to sort of a reasonable limits that can be demonstrably justified in a free and democratic society. So there is a weighing of the interests and the risks and benefits. And my last slide. So in conclusion, I was talking about the potential for future human rights to be developed in this area. And one that comes to mind that to date has mainly been used in the environmental sector, and that is the rights of future generations. So when you're talking about gene editing, for instance, and if one day we were to do germline or hereditable editing, would there be some sort of intergenerational monitoring necessary in the best interests of the child? Would we frame those interests of non-yet existing, non-yet conceived, non-yet implanted embryos and so on as a right of future generations. So there is potential to work in this area. And I conclude with the a statement, Article 2 of the Universal Declaration on Bioethics and Human Rights that seems to allude to this possibility of one of the aims of the Universal Declaration on Bioethics and Human Rights to safeguard and promote the interests of present and future generations. What I'm trying to say here is that if you thought we had incredibly difficult and complex discussions in the past, I think a lot more is still to come. And as long as we keep the primary interests of children in mind, I'm really looking forward to working on the Pediatric Atlas. Thank you.
So I think our next speaker is Helen Firth, University of Cambridge, is that right?
Yep. Good afternoon, and I'm very happy to join you now from Cambridge in the UK. I'm a clinical geneticist at Addenbrooke's Hospital there, and in my brief talk this afternoon, I'm going to talk to you about the placenta and also consider issues from a rare disease perspective. So if we start off with the placenta, typically information about the placenta, such as its weight or description of its appearance or pathology reports relating to it, resides in the mother's healthcare record. But in respect of the developmental and pediatric cell atlas, especially in an ethical context, I think it's really important that we consider and review the developmental origin of the placenta. So the placenta is derived from the fertilized egg, and you can see that at the 3-cell stage, where you've got there are 3 cell types, but even on day 5 at the 60-cell stage, the trophectoderm is the start of the developing placenta, and that will go on to form the placenta. But the key thing is that this placenta shares the same genomic DNA as the fertilized egg and the developing embryo. And so therefore, genetic studies of the placenta should take account of the genetic identity with the child that was supported by that placenta during prenatal development. And this is somewhat different to the legal framing for decisions about the fetus, because when fetal tissues are studied, usually it's the context that that fetus didn't go on to become a living child, whereas with the placenta, fortunately, oftentimes and usually it does result in a live birth, and then the genetic identity of that placenta with a living person is important to respect. And I think that will become relevant when we think about governance, particularly in records data that is held for long periods of time, potentially beyond the age of 16 years. So that's just a brief digression to think about that context, and now going to focus more on rare disease and some of the lessons that we can perhaps learn with respect to the Pediatric Cell Atlas. And I wanted to start by talking about Decipher, which is a project which we run from the Sanger Institute in Cambridge, which it has the mission to map the clinically relevant variants in the human genome, and do that by associating rare variants with clinical features of disease. And it's a big global initiative which has been running since 2004, and we take in a large number of data sources both from disease and control populations, so that we can present and contextualize a variant on the basis of clinical and population data to help with the interpretation of variants, and also to help with discovery of new causes of disease and the new functions and roles for genes, and for genes with existing function, whether there are additional diseases associated with a subset of variants within that gene. And we also share data with other organizations doing similar activities through the GA4GH matchmaker. So what sort of data does Decipher share? Well, we share these little cartoons of the genes, and this is a cartoon of the CDK13 gene. And you can see that actually all of the clinically variants are here focused in the protein kinase domain. So here's the gene with its exons. This area has extreme constraint on missense variation. This area is highly constrained, and that's the domain where the clinically relevant variants are clustered. We can also, because we collect morphometry and milestone data in association with those variants, produce these little ideograms, these little centile charts for height, weight, head circumference, and developmental milestones. So these are gene-specific centile charts, and centile charts obviously are a key tool in pediatrics to ensure whether a child is growing or developing according to expected norms. And one of the sadnesses faced by many families afflicted by genetic disease is that because of the child's genetic constitution, they're not destined to follow the usual norms, but they will have their own set of parameters which they would, a trajectory that they would usually follow. And by developing a set of gene-specific centile charts, we can have and present tools that can be specific to the expectations for that child. So they could be reassured that they're growing very well for their condition, rather than being continually told that they're far too small or too thin. So we can also put this material together to form composite faces, and we also collect these cases together and share the aggregate knowledge that enables the data and the value of and the knowledge generated to be shared without undermining the privacy of the participating individuals. And we also participate in matchmaking, as I mentioned previously. So what relevance does that have to the pediatric cell atlas? Well, I think the key points I was wanting to try to get across with the issue of identifiable data or non-identifiable data, and the great value of aggregating data such that non-identifiable data that doesn't contain individual genetic sequences can be shared openly without compromising privacy and create a public good to benefit humanity. Whereas identifiable data containing individual genetic sequences, I think, is best preserved and held subject to restricted access and governed by a data access agreement. So I mentioned before these centile charts. The individual measurements applying to a specific individual can't be deduced from these charts, but they have huge benefit in providing care globally that's tuned to a child's specific diagnosis. Similarly, this face was composed of 11 discrete images, but none of the 11 participants that generated that composite face can be identified from this image. And yet that image has critical value to people seeking to determine whether a child has this condition or not, and that can be of benefit diagnostically. So the mission of the Human Cell Atlas is to create comprehensive reference maps of all human cells, the fundamental units of life, as a basis for understanding human health, diagnosing and monitoring and treating disease. And I hope that's illustrated how aggregate data achieves that mission of a global reference and a public good. But restricted access may be necessary in some contexts, particularly those contexts that contain individual sequence data, to respect the privacy of individual participants. So, thank you.
Thank you, Helen. I think our next speaker is Dimitri, also from the Center of Genomics and Policy at McGill. Slides look great, Dimitri.
Well, hello, everyone. My name is Dimitri Patrinos. I am an academic associate at the Center of Genomics and Policy at McGill University. And today I'll be speaking with you about the issues of consent and recontact in pediatric research. Next slide, please. So as an overview, I will be describing the process of informed consent in the pediatric context, as well as what happens when we want to continue to use samples and data that we've collected from participants once they reach the legal age of majority or the age of maturity. Next slide, please. So to provide an introductory overview of informed consent is essentially obtaining permission from an individual for them to participate in research. And informed consent must satisfy 3 requirements. It must first be voluntary, so the decision to participate must be made freely by the individual without any form of undue influence, coercion, or deception. The decision must be, as in the name, informed, meaning that the prospective participant was provided with all information relevant to participation in the research project. So this relates to study design, procedures, risks and benefits, implications of participating in the research. And finally, the individual must have legal capacity to provide informed consent. So within the pediatric context, this is slightly different given that minors generally lack the capacity to provide informed consent. So in this scenario, we have what is known as proxy consent, whereby their parents or legal guardians will consent on their behalf and provide permission for their child or the child under their guardianship to participate in the research. However, general ethical guidelines do require that children be made a part of the decision-making process, and they can do so via providing their assent. So this is an affirmative agreement to participate in the research, and generally a minor is able to do so when they're old enough to have a general understanding of the research and its implications. Next slide, please. So as an example of the informed consent process in the pediatric research context, I've chosen the rare disease context, given that most rare diseases are child onset or pediatric onset conditions, and parents and guardians have a very important role to play in the informed consent process for rare disease research. And indeed, there's very strong parental and patient engagement in the rare disease community. However, technological and genomic advances have made the informed consent process for rare diseases perhaps slightly more complex than for other types of research. So for example, certain phenotypes for rare disease patients may require collecting audiovisual data, so facial imaging or videos. Often there is the possibility for familial participation in the research, so collecting perhaps biological samples from consenting relatives such as parents or siblings, and also data sharing, so the ability to share data widely to create meaningful research. in the rare disease context. Next slide, please. So I have an example here of model consent clauses for rare disease research developed by the Global Alliance for Genomics and Health. And this was developed with the collaboration of representatives from rare disease patient organizations. And these model consent clauses do account for some of the unique features of rare disease research. So as I said, the collection of audiovisual data, familial implications, data sharing, and so forth. Next slide, please. So another example here are of some audiovisual tools that we've been developing in conjunction with patient representative groups. So given that the rare disease context will often involve the participation of minors, we have here the development of audiovisual tools that can facilitate the informed consent process and make the information more accessible and more comprehensible for minors and for parents, so for families altogether. Next slide, please. So I mentioned a little earlier that to be able to provide informed consent, an individual must have legal capacity. So at what point does an individual actually obtain this capacity? At what point can they provide consent autonomously? So across jurisdictions, we have two approaches for determining the capacity to provide informed consent. We have first a majority-based approach, which relies on the individual attaining the legal age of majority in their jurisdiction. And we also have at the other end a maturity-based approach. So this approach does not rely on a legally fixed age, but rather the individual's competence to understand certain information and make decisions accordingly. So from here we have the mature minor doctrine, and this is a legal regime that originated in medical care and recognized that minors can acquire the competence to make certain decisions for themselves before they reach the legal age of majority. So this originated in the medical care context, but has since also been translated into the research context. Next slide, please. So for pediatric research, what happens where we collect data and biological samples from minors, but want to continue to use them past the point where the minor reaches the age of majority or maturity? So we can think here of biobanking initiatives or longitudinal studies. So what happens at the point where we want to continue to use the data and biological samples or share them, make them available for other research? Do we need the minor's consent at this point? Next slide, please. So in short, the answer is yes. This is the maturity view in research ethics, and the ethical basis for this lies in the principle of respect for persons, respect for autonomy, that recognizes that where data and biological samples have been collected from a minor, when they reach the age of majority or maturity, they should now have a say or some form of control over what happens or what will happen with their data and biological samples. So there is some dissent in the literature, and I've provided some references here of some authors who put forth a dissenting view. And this has to do with the feasibility of attempting to recontact minors for their consent to continue to use their data or biological samples, as well as questions over the scope of parental authority and whether this, the permission that the parents provided for their child, for the minor, does this continue to extend into adulthood or into past the point where the minor reaches the age of majority. However, the majority view continues to remain that where a minor has reached the age of majority or maturity, their consent should be sought for the continued use of their data and biological samples. Next slide, please. So when we do recontact the minor, how do we obtain their consent? How do we consent them? So we have three general approaches here. First, we have a new consent. So we ask them to provide a new consent for the use of their data or biological samples. We have next, a notification with opt-out approach. So under this approach, we inform the individual of their right to withdraw, so the right to say no to the continued use of their information, their data and samples, but no formal consent is required. So they're informed of the right to say no, and they can say no and withdraw if they wish to do so, opt out if they wish to do so. And finally, or rather exceptionally, we have the possibility to obtain an ethics waiver. So this is for special circumstances. I bring here the issue of attrition, which can be a major issue for longitudinal studies especially. And I've provided the example of the Canadian Health Measures Survey here in Canada that had enrolled minors, but did encounter very high non-response rates when they attempted to recontact these minors for the continued use of their information. So depending on the ethical guidelines in one's jurisdiction, if certain criteria are met, there is the possibility of obtaining an ethics waiver so that the researchers do not have to recontact and reconsent the minors from whom data and biological samples were collected. Next slide. So I provided some references here of the different ethical guidelines that sort of outline the process for informed consent and recontacted pediatric research. And thank you all very much for this opportunity. Thank you.
Wonderful. So our next speaker will be Vasiliki Rahimzadeh from Stanford University.
Thank you, everybody, and thank you to the organizers for inviting me to share the results of some empirical work we did to establish high-level policy points to consider in the responsible sharing of pediatric genomic and associated health data. And I'll as well be offering some thoughts on how the results we found really come to bear on the future endeavors to move towards a genomics-enabled learning health system whereby we apply data generated from the research context to advance clinical care and in turn use the clinical encounter to inform future research directions. And so my plan for the afternoon is really to briefly describe the process for developing the framework, as well as discuss some of the confirmatory results we found during a Delphi validation study of the framework with Canadian stakeholders specifically. And I'll actually spend a bit more time on what we didn't find in that study, which was agreement on issues related to perceived informational risks of sharing genomic data in relation to how they should be made accessible, as Helen actually touched on a bit in her talk. And these negative findings, as I'll call them, really suggest areas of future ethical, legal, and social research, as well as some of the technical challenges where further research could really substantially shed some light on how to move forward. And so I'll start, I'm an active member of the regulatory and ethics work stream of the Global Alliance for Genomics and Health. I'm currently based at the Stanford Center for Biomedical Ethics. And a few years ago, before the Global Alliance's restructuring, we were organized around special interests task teams, of which one was a pediatric task team. And so there was a representation of clinicians, genomic researchers, ethicists, and legal scholars from North America, Europe, and East Asia on this pediatric task team. And we tasked ourselves with conducting a consensus committee process to really synthesize some of the key priorities, practices, and values guiding the responsible sharing of genomic data involving children. And so I conducted part of this study as part of my doctoral dissertation work, in which I started off posing this question to the literature and did a systematic review of reasons to develop some of these initial policy points to consider. And what you see here are some of the 4 thematic categories that came out, both from that review as well as from consensus committee meetings with this pediatric task team. You'll see these high-level policy points situated around children's involvement, really based on the human rights framework that Professor Knoppers explicated a little bit earlier, around best interests, around their decision-making, participation insofar as they're developmentally and appropriately able to do so, around the authorization of parental consent, so how parents should really be informed in transparent ways about the ways their children's data will be used, not only now, but also potentially in the future. And this balancing of benefits and risks, as well as how genomic data are being protected and released. And what was most striking, and I'll explain in the next slide, during our Delphi validation study, is that we didn't receive consensus on these 2 statements that are indicated there in the blue about how anonymized or even aggregate pediatric data should be made available, and then this ethical obligation to allow parents and families the opportunities to share their data, not to oblige data sharing, but just to offer the opportunity to do so, was led to quite some contention and dissensus. And so in this Delphi study, which for those who are unaware of the method, involves structured communication and feedback across a survey, iterative rounds of surveying with expert panels. We conducted 3 rounds in which key stakeholders, again, similar to the task team, clinician scientists, bioethicists, IRB members, and clinicians participated in this 3-round survey study. And they rated each of those statements outlined in the KIDS framework on a 4-point Likert scale for their relative importance, feasibility, desirability, and confidence. And what we found were 10 policy statements plus 1. So one of these new statements emerged in the course of the Delphi on the return of actionably finding secondary genomic results. And there was at least 70% agreement across 2 contiguous categories of these ratings. So that's how we got to these 10 validated statements. And the 3 remaining are those you see here in blue, about that anonymized pediatric data should be made available, publicly accessible ways, that identifiable pediatric data should be coded and made available in controlled ways, and this opportunity to allow families and children to share their data. And so we also conducted a spread analysis, which is used to measure polarization. And these are the 3 that really yielded low consensus and high measures of polarity. And in these findings, we were really able to show that the potential for re-identifiability mediated by the openness of the access regime really shaped these risk perceptions among the stakeholders we engaged. And, we found that clinician scientists and ethicists tended to emphasize, the potential for the clinical benefit that could possibly result from data sharing. And so I think this is a subtle yet important point, that the ethical, legal, and social issues of data sharing are perhaps best understood and contextualized and articulated as trade-offs rather than as static principles. And so here I've sort of summarized some of those. And the first one involves the tension between research protection and promotion. So children constitute a vulnerable research population across, many jurisdictions and as codified in international conventions, really whose inability to consent to research and to some research and clinical care decisions warrant additional ethical protections. And so this tension is always between protection and promotion, and as in the case of the data intensive sciences, around data protection and promoting pediatric data intensive research. And you know, as Professor Knoppers mentioned, it's that children encompass a population to which they deserve to be beneficiaries of the fruits of knowledge that emerge from science. And there's an element of time in this as well, and the idea that the accrual of information risk over one's lifetime is theoretically longer for children than they are for adults. So we know there's an inherent privacy utility tension in this. So where genomic sequence data and other related health sources are linked together are especially useful for research, but can incrementally raise the identifiability risk quite dramatically. And so considering these tensions while developing the KIDS framework, the working group adopted the position that security standards for such data sharing should correspond to the nature and quality needed to generate the best available clinical interpretation of the child's medical presentation. And we heard extensively just recently about obtaining recontact and reconsent for the continued use of adult data collected when they were children. And this is something widely supported in the literature, again, in theory, but to date has really posed some formidable challenges to execute logistically, especially in longitudinal studies, also with conflicts due to unable to follow up with adults, again, once they reach this age of majority. And then finally, you know, really the data protection landscape is continuously evolving. At one end to meet the increasing demands of a data-rich field, but also, you know, as a political arms race for data play out both at the continental, the national, and even now at the regional level. And so it's, you know, as a result of different data protection approaches, so some like the GDPR that are very generalist and widespread, they considered, you know, personal data as encompassing lots of different types of data, let's say in relation to the United States, where we regulate data based by sector. So these are different approaches that are really adding and complicating international data flows of genomic data, including from children. So in the very few moments I have left, I'll leave you with this workflow diagram that maps some of the challenges I spoke of onto what I envision a genomics-enabled learning health system might look like, acknowledging that we have quite a lot of work to do in ensuring that at every stage of this pipeline, that children of diverse genetic ancestry are represented equitably in the data sets we collect, and that the benefits of the data science we create are really distributed fairly and equitably across those populations. I'll end there.
Okay, wonderful. So I think on to our last talk before the panel discussion and Q&A. And last, certainly not least, will be a talk from Deanne Taylor, who probably needs no introduction given her leadership in this space, but is located at Children's Hospital of Philadelphia. Deanne?
Thank you very much. I'm going to talk a little bit about postmortem pediatric sample collection and the LC issues around that. Click on this. So mostly the previous talk by Besa was great because it kind of introduces us to the overall problems of sharing pediatric data and safeguarding it. And in fact, what we'll be doing when we collect pediatric postmortem data and tissue samples is really safeguarding family interests as well. The donor is deceased, and future privacy concerns are really focused around the family and the communities where that data is being collected. So there really is potential risks in the research collection are really overlapping with those from liver donor tissue collections. But again, the risks go to families. So for example, collected molecular data associated with the genetics, you could re-identify donors and donor family identity. And some communities may see that as an invasion of privacy and safety in terms of re-identification. Collected data storage, security and sharing, risk of re-identification if we have an open access data sharing model with postmortem data. And this has to be addressed in terms of authorization and parental consent. And of course, the unknown future risks of samples. If we collect samples and we put them into a biobank, how long are those samples going to be kept? And how does that provide future risks for families and even family members who aren't even born yet? Do they have any say in this? And obviously that kind of thing is hard to trace, but how can we safeguard the data to a point where the risks to those populations are minimal? So there's a psychosocial risk for donor families in the grieving process, and also, of course, and I don't cover that in explicit terms, but there's also social community implications for donating tissue. And we try to minimize the risk for donor families by using well-trained personnel, genetic counselors or grief counselors, and also, people who do clinical trial management or other types of management who have expertise in discussing this kind of subjects with parents and navigating them through the authorization and consent processes, and offering them parental guidance. There's often parental boards or parental groups that serve in hospitals as kind of a boards of people that can advise other parents on research concerns or privacy concerns, and also professional support services that the organizations offer. However, not all of tissue collection happens through hospitals. We've discussed in earlier breakout sessions today that some of these can happen through, like, for example, medical examiners, or in other countries, maybe through other sources. So there aren't always those resources available to parents. And often parents operate from a community or, or from a historical point of view, how research interacts with their community. So there is a psychosocial risk there for donor families and how they respond both to the loss of their child, but also donating that tissue. And then there's a question of recontact, whether we're able to recontact those donor families, or if we even want to as researchers, to obtain, for example, family of subject histories not known at time of death. And this is actually pretty common. People always say, well, we should have thought of that before we started the study. And hopefully people spend a lot of time thinking about this, but there's always something that could slip under the radar. So it could be a benefit to investigators when the tissue collection phase can't access some medical data, which often happens, as I mentioned, but there's a risk because you're recontacting the parents after the death of their child, where they're trying to move on, opening up a lot of those wounds when you start asking them about the child. And there's been publications that have mentioned this as a significant impact on the parent when you're recontacting them about it after the death of a child. So if necessary for the study design, the recontact should be discussed with the parents as part of the authorization process. And then there's the overall perennial return of results questions. And there's been a lot of discussion in previous breakout sessions about the need to acquire genetic data along with single cell data for the good of the study. And that, of course, introduces possible discoveries in the child's genetics that may have implications for the family at large. For instance, a BRCA diagnosis, or some other diagnosis that has a implication for the future health of the family. And so there's a benefit of returning those individual results potentially to the family, but has to be carefully considered with the risks back to the family. For example, one thing that has been brought up many times is cryptic parentage. You may have a child or even a family who's unaware that the parentage of a child is not what it was expected to be. And those return of results can interfere or can actually introduce knowledge that people may not want to know. And there's also psychosocial impact of recontact, like as I mentioned, reminding the parents of that loss. And also interpret, whether or not you can actually interpret the clinical data from a research perspective, because when we do research, we're not clinical diagnostic people. So you would have to involve clinical diagnostic people to perhaps validate the return of results. You don't want to give somebody something that you saw based on single cell data, for example. You'd want a clinician to come in and say or retest a sample, for example, and to display that as a valid result. And if you're planning on recontacting, studies really need a plan to operationalize that recontact. You have to have resources for that. You have to have, again, bring in clinical people as well. And you should have specific guidelines on what to return. There was an NHLBI working group, for example, that presented those guidelines. And also there should be some clarity regarding return of results in the authorization or the consent that you work with the parents on when you first acquire the tissues. So in general research use, most of the postmortem tissue and authorization protocols that people offer to the parents is usually in most cases, or the most optimal cases, a general research use case where, I mean, some studies could say we're only going to limit the work with your postmortem tissue only to the study of pediatric cancer. But to be most useful, general research use, you know, having a broad consent in that sense is very useful. But you could also add restrictions, and I don't think I added disease-specific restrictions here, but I should have. But also there's contact of families, again, as I mentioned, whether or not this is going to be used for profit or allowed to be used for profit research. International use could cross borders, and this is often baked into some countries' legal structures, or whether or not data can be passed over borders. Whether or not biosamples can be indefinitely stored and used over long term, and other considerations that you hear, especially cell line generation. I mean, can we generate cell lines from tissues in children and study organoids? Well, that's almost immortalizing the samples, and we know the story of Henrietta Lacks and the HeLa cells, and how, you know, the family wasn't quite aware that this was going to immortalize their relatives' DNA. And so these are kind of considerations and discussions that have to be both done in the research group before it goes off to the parents for authorization. And this is the last point is something that is especially important for living donors, but we haven't really talked about this as a community, is whether or not postmortem donors' data can be withdrawn. And if that was going to happen, how do we control children's data, children's postmortem data, to allow for withdrawal at a later date, or should we? And that maybe should be baked into the additions to the consent or authorization. Tissue collection, when tissue collection is allowed, now this, I kind of summarize across the globe, and I'm certainly not an expert. I have not done the due diligence to do a complete global survey of postmortem sample collection in children, although that probably should be done, so we can understand really what the rules are, if there are anything specific for children. But generally, next of kin authorization is almost always required for research use that may reveal identifying information. There are some protocols in different states and different countries where if it's completely de-identified, it might be allowed to collect that data. But again, genetic studies could identify those people. So we have to revisit those. And often there's use of research review boards like IRBs per project are almost always required. Not always, but in most cases they're required. And some jurisdictions want to remove all identifying information from postmortem samples, or as much as possible, which kind of interferes with the research process, depending on the jurisdiction. There's also time limits on how long data can be kept, and that may include tissues. There could be a 10-year moratorium, for example. I think Denmark has one of those, a 10-year moratorium on data collection for postmortem samples, or data retainment for postmortem samples. So there is a lot of change from country to country when you look at these kinds of rules. Long-term biobanking, I don't think has been, I mean, people have studied biobanking in pediatrics a little bit. There's a couple of papers out there, but I'm not citing them here, but you can find them if you look for them. But essentially there aren't a lot of cohesive rules and regulations around biobanking from country to country, or even within a country. In the United States, for example, different states have different rules on data collection and sample collection that can vary, even though there's a federal, in the United States and also in Europe, deceased donors are not considered quite the same as living human subjects. So there's also social implications. Vulnerable communities of diverse backgrounds have to be supported by ethical reviews. We have to make sure that when we do pediatric postmortem collection and biobanking, that these communities are fully engaged. There's also religious, traditional, and social values associated with postmortem data collection, especially for children. And I don't think there's been a full review of those, especially when it comes to single cell research, which really requires immediate tissue collection. And implications long-term for sampling diversity. All of these considerations above have a long-term impact for getting a diverse representation in medicine for different ethnicities and different ages. One suggestion was parental review boards that may be chaired and led by parents in the local community could assist in understanding the implications of research donation for those parents. And there's also groups and communities that may require focused work with local medical institutions, community groups, et cetera, to assure participation, to assure the representation and scientific advantages to that community, balancing the needs of the family who want to donate and the community concerns with donation to the advantages to that community and the living children and the future children to the fruits of the scientific research. So I'd like to stop there, and I'd like to also thank Melissa Von Dran at the National Disease Research Interchange for some statistics and use and help with my presentation. Thank you.
Jonah, there are some questions in the chat, and people on the panel, don't be shy putting questions in the chat as well. I think I'll take the first one, which had to do with the international sharing of sequence level data from pediatric samples. The question came from the US, but I think it's a universal question. My answer is not very positive. As you know, the General Data Protection Regulation in Europe, which was meant to cover Europe and the EEA, actually has had a greater, what we call in law, extraterritorial effect than I think they ever could have imagined or dreamed of, with the result that international collaborations automatically feel obliged to meet the very strict standards of the GDPR. And in the GDPR, health data and genetic data are considered sensitive data. So they've created a special category and created additional obligations. So you will find that the tendency is to go towards controlled access, managed access, restricted access, whatever your country calls it, which usually involves having some sort of data access committee review, and also some sort of data protection requirements, meeting certain data protection requirements in an assessment. Now, I don't think that all hope is lost. I do think that there is potential for, at a minimum, making us, with some of the techniques that Ellen was describing, making minimum data available, because what we need to know is not just about one child somewhere. The interest lies in, having a map that is not only representative and international, but also open so that you know where these genetic sequences are, these alleles are. And my techie friends, IT friends tell me it is possible to make such a open database available without running into the forever threat of re-identification. And when I say re-identification, I mean a reasonable likelihood of re-identification, because you can always throw out this hypothetical, what if one day somewhere some hacker does something, which I don't think is an ethical way of approaching it. And finally, I would just say some countries, like the UK, for instance, has legislation it would be is in the criminal code. It's criminal to attempt to maliciously or intentionally re-identify an individual. And I think that's one route we should go, is just to get it out there and say this is verboten behavior, and then we don't have to make all our policy based on a potential of something that really should be criminal. But there must be other points of view out there.
Deanne, do you want to speak? You put a nice comment in the kind of speaker thread about some kind of current efforts around this, and wonder if maybe it would be helpful to raise those, or if others have examples of how different groups have approached this or are currently trying to approach it.
Yeah, there are several efforts out in the wild using a software called Beacon. I'm not sure how much it's being used right now, but for a while it was it potentially can allow you to query a large cohort, like if you have a biobank or a sequence biobank, to ask if a certain cohort has an allele and bring that back. And actually Kids First itself, which is a project run by the Common Fund at NIH, they have a variant warehouse that won't explicitly tell you what child has that allele, but will tell you if a certain birth defect cohort has the allele. You can look up that allele and it'll tell you what cohort it comes from, but it doesn't give you any alleles around it potentially. So you don't know, you don't have the actual genotype of the individual. You just know whether or not a potential deleterious allele is available or is showing up in that cohort. So there's a couple of different solutions for this, both summarizing, de-identifying cohorts, and Nomad, of course, and Exact kind of do that as well. And then there's the ability to go and just actually query an actual cohort and say, does anyone in your cohort have an allele? And you send out through an API and get back a ping saying, yes, this allele is present.
Yeah,
So
that's just been standardized, Deanne. I think in the last GA4GH meeting, they've come up with an international standard exactly for that. And Ellen mentioned the matchmaker, and the first step of a matchmaker is exactly that. Like, anybody out there seen anything like this? So that is more a discovery. It's not a re-identification, right? And that should be encouraged and permitted. And then when you want to go down to the clinical case or person or whatever, then of course you run into your privacy, consent, and other issues. But that discovery potential of open access databases and using a beacon, and still the richness that you can get by being able to find that allele
I think very much that it's an issue of proportionality, and sort of in Decipher we aim where we can to seek consent to share a variant with the associated phenotype when that candidate variant is the most plausible cause of disease. And then for non-logged in users, they can't tell the global location of that individual. And so, we're not revealing where they live. So it would be very difficult to say that this individual is the person who contributed their data, because you would be thereby inferring that they were perhaps the only person on the planet with that variant, which is very unlikely. So I think it's a question that the moment you start sharing large amounts of sequence, strings of sequence, then it does very rapidly become identifiable. And particularly in regard to children, I think there's the issue that by triangulating various incomplete data sets that none of which can you independently identify someone, but if you triangulate this 2 or 3 data points, then you actually can create a web of information which allows you to identify people, and there have been several papers illustrating that. And I think the potential for that is likely to grow over time, and that's a particular issue when we're charged with responsible behaviour towards samples from young children.
So I think there was sorry, go ahead, Bartha.
No, it's just that we've been looking at what privacy commissioners around the world would consider to be the potential for a reasonable likelihood of re-identification, because of course we can't protect 100% either in the present and even less into the future. And looking at what they've come up with, they're now beginning to go from risk qualification, i.e., is there a reasonable likelihood, which is something I can understand, and it's a proportionate approach, to a quantification approach on accepting that in any given society, especially with changes in technology, they're accepting from up to between 5 and 9% chance in the future as still being sufficiently privacy protecting. And I think that's what we should do, because it is also in the interest of children to be in databases. They are a population. They're not just an individual child, and they have their own diseases, their own conditions as a population. And that contribution to improving the quality of care, Vaso was talking about learning health systems, bringing the research back in and into the health system. And if we ever get there, we need to code and encrypt and be able to find so that enriched information can feed into improved clinical care. So I think we over sanctify, or whatever, there must be another word, sacralize, who cares, the whole like re-identification issue, and it blocks the greater public interest that Ellen mentioned in having good quality science for children specifically that will benefit other children in the future, with a risk, yes, maybe one day, of re-identification.
So I see there's a number of new audience questions, so maybe trying to get to a few of these. So one came from Heather Etchvers, and a follow-up on the concern from various groups about donations from prenatal tissues and the possibility of re-identification, not just of the tissue donor, but also the mother or the families. So maybe a curiosity here, and kind of putting very, very rare variants aside, but thinking about kind of more general and expected variation in otherwise healthy tissues. Any thoughts on the possibility, the likelihood, how we're thinking about frameworks or guidance for the community as this work proceeds? Maybe Vaso, do you have a thought on this one? And your talk spoke to some points.
I think Helen was about to jump in, and maybe I'll sign myself up for Bruce's question about IRB expertise. I'm doing a bit of work on that currently. So Helen, please.
I was really just going to comment that, I think that those are particularly sensitive contexts, and I think my initial thought would be to try to work actively to see how it's possible to hold, to pool small amounts of individual data and then share an aggregate that isn't possible to deconstruct into individual data, because I think you can still derive huge benefit for knowledge and drive research forward if you're careful and very nuanced in the way you don't necessarily have to provide the absolute raw individual data, but if you provide it in such a format that you can't deconstruct it into the individual component parts, you can still provide data that has huge benefit and can drive the science forward. So that's a context where I would be working particularly hard to try to walk that tightrope and get that balance right.
So unless there's additional thoughts here, Vaso, do you want to?
Yeah, no, Bruce, I appreciate your question about IRBs, particularly because it's something we've heard anecdotally from members of various work streams through the Global Alliance, namely the technical work streams, articulating that they're these studies and the IRBs that review them really lack some of the data protection expertise and sometimes even the computer science expertise to make risk assessment, judgments about how risky a protocol is, or, whether there might be some downstream opportunities for non-compliance with the data that's generated as a result of some of these protocols. And so that is a project I'm working on through another consortium that's asking IRBs, do you think you yourself have the requisite expertise to evaluate data sharing plans, data management, processes and practices, and I guess I'll give you a bit of a spoiler alert, is that it's pretty widely, as far as our survey data is concerned, and it has a rather good sample size, is that among American HRPPs, so these are of the human subjects rights offices, are claiming that they have adequate expertise in some of these areas. And so I think that's the next big challenge in equipping, oversight bodies with some of that expertise and some of that training and knowledge. And, the downstream implications of having that expertise can't be understated, because as soon as you have an IRB approval, that really carries down to data access committees, which sit a little bit downstream of this pipeline, in which they are the essential gatekeepers of database data, secondary use of data, and are matching what was consented for and approved from an IRB standpoint, and ensuring that compliance carries on to secondary use. So really, this is almost a domino effect, and IRBs, research ethics boards, research ethics committees, wherever you are in the world, and data access committees indirectly work in concert together to assure, a data management pipeline and data use are used as participants intended for them to.
So I think there's maybe one additional thing that I'd add on to that, Baza, which is the importance and the engagement with patient communities and kind of the public in general.
Absolutely.
In particular with rare disease, many of these groups are among the most motivated to make data accessible as quickly and as openly as possible.
Exactly.
Working with them while also maintaining privacy, and, to find that balance and the education, I think is both a really exciting and really important opportunity.
Dimitri, can you answer that last little question about what do you do when you want to withdraw? What about withdrawing pediatric data consent? How can you do it if it's been made publicly available already?
Yes, well, I'll go very quickly because I see that the time has run out. I'm presuming that they mean that it's publicly available in aggregate form in a publication or something like that, or an abstract. I think usually at this point, once the data has been shared or processed, it's not really possible to retrieve it. So I think at this point, there are limitations to the recontact reconsent process in general, and not only for minors upon reaching the majority or maturity, but in general recontact processes. So at this point, usually what we see in consent forms is that the participants are informed of this possibility that if we share your data is processed or handled, it's not retrievable at this point. So it's disclosed this possibility to them, and they can make an informed decision accordingly once the information is out there.
Correct. Yeah. Jonah, are you closing the session?
I'm happy to, but maybe just to thank all of you and the other panelists and everyone that participated. The questions were fantastic. Anything to add, Bartha?
Thanks everyone. Presentations superb. Thanks a lot.
Thanks. We'll now take a 10-minute break. So we hope that you join us back here at 1:41 p.m. Eastern Time for Mazlifa Hanifa's keynote presentation. Hope to see you then. Thank you everyone for a great session.